Today we are so thrilled to welcome Dr. Wesley Weber. Dr. Weber recently completed his doctoral studies at the University of Alabama, and today he will present some information about his dissertation study where he analyzed implicit stereotyping among graduate students in mental health fields. We are so excited to have Dr. Weber share his insight behind the mechanics of event-related potentials so we can better understand measured brain responses. So Dr. Weber, thank you so much for being here. And just as a reminder, we will present for about an hour and then we'll have an informal discussion at the end for about a half hour, which you are all encouraged to come if you can. And if you have a question, just drop it down in the chat box. And I will also post a link to the CE certificate survey about 10 minutes up past the hour. Thank you. So whenever you're ready, Dr. Weber.
Great. Thank you so much, Emily. It's a great introduction. As Emily mentioned, I just recently, feels like recently, maybe that's a subjective statement, finished my doctoral training at the University of Alabama. So this is my second semester teaching at Tennessee Tech. And as Emily said, I'll be discussing some data from my dissertation today that you read about in the manuscript, and also giving you kind of some broader information to maybe better help understand the methodology and that kind of thing. So really look forward to talking with y'all today. Go ahead and move through here. So today I want to first tell you a little bit about my counseling and neuroscience research background that really allowed for the study that you read about and that I'll be discussing. Also want to talk about what I say is the value of experimental EEG and ERP research. So the sort of experimental designs that I used in what you read. And then in that context, I'll talk about the study itself and present that to you today. Sorry, let me get my screen in better order here. Okay, so I feel like something I want to share when I present with the group who I don't know to what extent you have read these kind of designs that you read in my study. I want to say I am a counseling researcher. My PhD is in counseling, so I almost want to say, in your introduction to me, I don't want you to think I look for ways in my research to put people in front of a screen and have them make a single response 502 times. Like, you know, that's a certain method that today I want to hopefully kind of convey to you has value within neuroscience research. But I certainly have been fortunate to get a lot of great traditional counseling research experience in my PhD program. But what really has contributed to my knowledge that led me to produce the study that you read about was the ability to collaborate throughout my doctoral training. So within our department at the University of Alabama, when I was there, there came to be this focus on neuroscience, specifically educational neuroscience, as this new kind of field of bringing in really methodological experience with neuroscience into educational research. So it's really becoming a field. There are multiple programs in the US at least that have this focus, and UA became one of them. And so I was able to take classes alongside these students and faculty, and consistently gain really empirical research experience through working with them, these new faculty that came in with neuroscience training, as well as some of our counseling faculty who were starting EEG projects as well. So really consistently for a period of about 6 years, I was kind of around this, whether it's through training classes or just applied research experience. This was from the point at which this began at UA. So it's also really exciting for me as a counseling researcher to kind of see the ways in which these methodologies can be integrated into a field and really have these discussions about what does this lend us in addition to the methods that we're used to. So that's happening within education, and kind of a connection point I've had with many of those that I worked with was that I'm interested in the same thing within counseling. As I've listed here, there's come to be a lot of great resources with this program. So it's now a PhD with a concentration in educational neuroscience. As of last year, there's now a bachelor's of science at UA in educational neuroscience, and there's now 4 neuroscience research labs that are run by the new faculty that have been brought in. Okay, so as I mentioned before, I'm very conscious of when I present this type of method to counseling people, even those who are maybe interested in neuroscience. I don't know what kind of familiarity you have with reading the kind of method that you read in the manuscript that I shared with you. So for me, my interest in neuroscience and its integration within counseling didn't really start with considering these kind of detailed experiments, computerized things with tons of trials. I started out interested in a lot of times reading more conceptual, theoretical pieces that would really frame neuroscience in terms that we're already used to thinking about, the kinds of constructs that we've developed in our field through the kinds of methods that we're familiar with. I think that becomes kind of it was a starting point for me, and I wonder if it may be a starting point for others. So for me, when I first would see methods like this, I'd feel like it would kind of drain the inspiration from me. I would ask myself, I'm struggling to understand, how this relates to the real world, things that I enjoy considering implications of neuroscience for. Well, now on the back end of all these years of experience that I've gotten, that I mentioned, I now find these methods, the same ones that kind of suck the inspiration out of me at first, to be the most inspirational kind of thing. So, I wonder when I'm presenting to a group if maybe there might be somebody in the audience who feels the way I did when I first was exposed to these kind of methods. So, I wonder what might I share that could convey where I stand with this now? How can I possibly now find this sort of method and its utilization pretty inspiring? Well, one thing that comes to my mind is knowing the things I know now about EEG designs, methods of analysis. If I kind of step outside of all that and think about just the bare bones of collecting EEG data, it's actually pretty astounding to me that we can develop methods that can tell us anything specific related to psychological constructs. So things that we can put words onto, kind of ways that we're used to doing this through our own methods. In a way, it seems like an absurd idea that this is possible. So some points that kind of that I'm thinking of when I say that are, we think about how EEG electrodes, when we collect EEG data, we're getting information from the brain, presumably, but the electrodes are separated by a skull, skin from the brain itself. So, that would seem to present some issues in terms of what's the information you're getting. Also, this scalp recorded EEG, to the extent that it is a reflection of brain activity, it's a reflection of a lot of brain activity. So we can't, tell the electrode exactly where to get its information from. It's a reflection of a lot of stuff. When we collect raw EEG, so just the recording we get during some period of time, it's also stuff that's not brain activity. So just a subtle movement introduces muscle activity into what's being collected. So it becomes non-brain stuff mixed in with brain stuff. Additionally, when we collect EEG data, moment by moment, activity in the brain is changing. So, with that in mind, how do we even define a construct? Since it's such a temporal thing, it's constantly changing. So these ideas, they kind of present a starting point where I'm amazed that we can create methods that can actually give us valid information over time. So that's why I say here on this slide, fortunately for decades, researchers using a particular method that I'm going to talk about, ERP method, event-related potentials, have been studying EEG responses in a really specific way. So in precise experimental conditions, and you'll see how precise when I'm talking about my own study, so knowing exactly what happened at the moment in which you collect EEG data. Also developing a method of creating as precise as possible data points that you actually analyze based on your recording. So doing this kind of thing in this experimental way allows us to compare brain responses across different experimental conditions when we know exactly what we've changed in the conditions. to see if maybe there are different responses based on subtle differences that we create in experimental conditions. And then in the same way, we can look at those sorts of responses across different participants. So we can think about the differences between groups and see if they show a different pattern of brain response based on their characteristics. So using this kind of design or these kind of alterations in experimental conditions for decades, ERP studies have replicated themselves. So showing that the same kind of responses and the same conditions are found repeatedly. This gives us validity to know that what we're looking at is true across different studies. And then we can tweak these designs to see what changes, what changed in the conditions we created and what changed in the brain response. So in this way, we really work towards developing a foundation, a literature similar to that which has emerged from the kind of methods we're used to. So we think about how constructs over time may be measured through self-report. We've developed pretty good ways of establishing validity with these kind of constructs, and we see kind of new constructs emerge that make sense based on previous constructs. So this same trajectory can be seen in EEG. It does look and feel a little different, perhaps a lot different than maybe the methods that we're used to, but this trajectory can happen, and in fact has happened in this method for decades. And to me, that's pretty amazing given the first statements I made about how EEG seems like such a messy thing, and it can be without these sorts of experimental designs and methods. So I want to, in that context, kind of tell you a little bit about what ERP data collection really looks like to be able to have that kind of precision that I just described. So whenever, for some reason I don't have my mouse, now I have it back, okay. So whenever data is collected, these are the EEG waves that are recorded during an experiment, again, where you've created exactly the context in which participants are doing whatever it is you've created. So this is the recording where you've got the EEG responses, and then every time something happens in an experiment on a computer that you create, you get a marker in the EEG data of what happened. So these numbers let you go back and tell exactly what happened and when in the data, which allows you to look at the EEG data at the moment that happens. So maybe a stimulus comes up on the screen, and then you see that the participant responded in whatever way that you've established for them to do. So this gives you that precision afterwards to be able to analyze the data in the way that you saw fit to do. So then after we've got the EEG data from an experiment, so you then have these markers. So let's imagine these are multiple time points where something happened in the experiment that's a certain condition that you want to get multiple iterations of. So you then have really multiple sample points here. And the ERP process, the way that we, again, get towards this precision in the midst of all the stuff that's happening in the brain, even in these precise conditions, is to average a bunch of these. So we want to average out the stuff that happens either just by chance, stuff that is not related to the process that you're attempting to measure. And when we average these, that random stuff averages out, and we see what's the consistent thing that's happening. And this gets us towards that kind of precision and validity. So we end up with something like this, which is called an averaged ERP waveform. And you can see generally, these things look a lot more smooth. We call that kind of clean data. We've averaged out random stuff, and we've got the consistent response in a given condition. Okay, so a way to think about this kind of research that gets us toward precision is we're just doing averaging on top of averaging. It's a way we kind of cut through the random stuff that happens in this complicated brain and data collection situation. So we can think about, these are different participants' data that we come up with in a given condition, so different subjects or participants. Now, oftentimes in papers, and what I'm going to present to you, we don't show individual subject data, we show the grand average. So the average across all people, all people in a given sample. So this lets us look at the common response. It can also let us kind of better see between groups differences as well. Okay, now this image can really let us see the real utility of EEG or ERP, really specifically ERP method. And this is even compared to other neuroscience methods. So a way to think about getting information from the brain in a valid way is to recognize that every method that's used has its own advantages and disadvantages. So I want to show you the advantages of this ERP method in terms of how it gives us information. So this is, again, we can picture this as an averaged brain response in ERP across different conditions when we've gotten this kind of average brain response in a certain condition. So this is the moment that a stimulus occurs, and this is the EEG response that we're looking at. So this trajectory here tells us that the different types of neural markers and things that we can isolate with this method really happen over time. So the first kinds of markers, and these things that are listed here are called components. These are things that maybe a given researcher might be trying to isolate because this is associated with a certain cognitive process in a certain condition. So these components, you'll notice that the first ones deal with initial sensory responses. So we process information first by kind of getting its basic sensory texture. And then as time goes on, these later components are more higher perceptual processes. At this point, we can categorize different items based on a certain task in an experiment. Around this point, and these are milliseconds at the bottom here, response selection occurs. We tend to have working memory encoding here, and emotional responses can occur right around here. So this gives us kind of a landscape of different processes that we can measure through setting up certain experiments and trying to isolate this given component and see, does it look different within different conditions? And what would that tell us about our processing of these different conditions? Or might the same marker look different in different groups? So for example, maybe a clinical population versus a non-clinical population on some basis. Okay, so this image is really, to me, it emphasizes the unique information that ERPs can give us. So, this is a sort of a standard experimental situation where a participant's looking at a computer, where you have that control of presenting, knowing exactly what you presented, knowing their exact response. Many times we can get unique information just by getting their reaction time to certain tasks. And so we can picture maybe along this continuum here that reaction time might happen. You might respond to a task right around here, like 600 milliseconds. And we can come up with some ways to infer processing based on that. So you all are probably familiar with implicit association test, where we can test bias based on reaction time. But what this ERP gives us is we can see all of these different types of processes that may separately contribute to the reaction time itself. So we get all of this information. So we can say the good stuff, the stuff that happens between a response and the stimulus that came before it. I also wanted to point out back to like methodologies and how I think many times thinking about sort of brain regions is maybe an easier entry point for many people. I think it was for me when I was thinking about ways to measure constructs. We like this image of localization or brain regions. ERPs kind of give us a different type of what we call resolution. So resolution is just precision. We can think of it as meaning that. fMRI does have a lot of precision in kind of the spatial domain of showing us where things occur, but ERPs have better temporal resolution. So we think about fMRI, we're not really thinking in terms of this precise timing. So in fMRI, all of the processes that you're kind of isolating are in a single time slice. Whereas here, we're actually getting precision at the millisecond level, which is not true of fMRI. So these kind of designs utilize that. We can, this timing becomes our way of sort of seeing these constructs, measuring certain constructs. And so we think in those terms, and it can give us unique information. So with all of that said, hopefully I've given you kind of a window into how ERPs can serve as markers of describable brain responses based on the conditions that we've created in an experiment. So we can use this fact to leverage our research to actually use the brain to answer specific research questions. So a question I found myself kind of asking for years, and it'll become the context in which I present this study to you, is how do we combine our existing counseling values and interests, the things that drive the kind of research, maybe more traditional types of research? how do we approach research with those same values, but best utilize the power of this kind of experimental neuroscience method? So I want to talk to you about the study that you read about as an example of that. So something that we have long cared about in the counseling field is the quality of our relationships with clients. So the multicultural social justice movements have really helped show us that deeply connecting with all clients across different culture groups is probably more difficult than we once recognized, and we've come to better understand that in time. So some theories that really help me better understand social cognition and maybe some complexities in working with diverse groups are dual process theories of social cognition. So this is to say that in our responses to other groups, particularly on a cultural kind of basis, humans are said to have both automatic and controlled components to their thoughts and feelings towards groups. In terms of the automatic kind of components, we may just kind of instinctively have a certain reaction just on the basis of noticing a certain kind of cultural characteristic that could be, like I said, feelings or thoughts. At the same time, in certain conditions, we can have control over those. For example, if we recognize it and we want to alter this immediate kind of thought, we can do that. In thinking about the ways that we study this topic in counseling research, I think it's important to note that we do use a lot of self-report measures kind of across the board. When we think about self-report measures in terms of this issue of controlled and automatic kinds of processing, it's important to note that self-report measures always allow for controlled processing, which sort of can be said to obscure our study of automatic processes. So even unknowingly, we may think about a question maybe pertaining to our feelings about cultural groups, and it may obscure whatever's automatically there. This is unfortunate because we do know that these kind of automatic responses have effects in the real world. So they're known to produce noticeable effects in our interactions with groups towards whom we're biased, affect our interpretation of different facial expressions of groups towards whom we have bias. And also within counseling, lately, this topic has become especially relevant because we've recognized the existence of microaggressions towards clients, which presumably may have something to do with unknown kind of automatic, implicit sorts of associations or feelings that we may have. So as I mentioned before, reaction time is a way to study implicit bias, so a tendency to have more negative feelings towards a certain group. But stereotyping is kind of a related but different topic in the sense that stereotypes towards a group can be either positive or negative. It's more of a tendency to just have an expectation just on the basis of knowing someone's cultural group. So this is the area in which it's what you read about in my study is really using a certain ERP marker as a way of studying implicit stereotyping. So what you read about was the N400 ERP component. So I want to tell you a little bit about this effect and how it can be thought to be a measure of implicit or automatic stereotyping. As I mentioned before, these ERP components are kind of the trajectory of it is they're usually noticed in a certain study where an experimental kind of condition is created and something novel is noticed within the EEG data. Then over time, future studies can use that knowledge of a certain effect to kind of study different things. So to help you understand how I've used it to study stereotyping, I think it's helpful for me to first tell you how it was first discovered, the kind of experiment. So this N400 is said to be all about congruence. So it's a reflection of an experience of a participant in an experiment of something that's congruent versus incongruent. And specifically, this moment in which a stimulus is experienced as congruent with its preceding context or incongruent with its preceding context. So to illustrate that, I want you to imagine an experiment in which participants are going to read two different sentences, word by word. It's these two sentences here. The thing that we're going to measure, the EEG response, is their processing of the final word that's underlined here of the sentence. So let's go ahead and think about these sentences. If you were to read the sentence, I take my coffee with cream and sugar. And thinking about how that last sentence, the last word feels to us, it makes sense to us probably. We're not particularly surprised by that ending. But this sentence, I take my coffee with cream and dog, to most of us, when we get to that word dog, it doesn't make sense. There's a feeling of incongruence there, whereas here there's a sense of congruence probably to us. So let's look at these EEG responses to processing these words. This is really just kind of fictional data to kind of illustrate this effect. So let's imagine this is the moment in which the final word is presented and the participants start processing each of these words. The blue EEG wave is the response to the word sugar, which, you know, we said felt congruent probably. The red is the response to dog, which we said likely feels incongruent to us. So we can see in these first moments of processing each of these words, there isn't any difference in brain response. So it turns out that, you'll remember I pointed out before that these early moments are a reflection of our processing sensory elements of whatever we're looking at. So these common elements are really just a reflection of the common task of reading a word, kind of identifying what came up on the screen, probably kind of identifying the color, the shape. So in those moments, there isn't any difference. But around here, which we could say is maybe around 250 milliseconds, so there are 1,000 milliseconds in a second to kind of give you some context. So 250 milliseconds or so, we see this difference. So this is the N400 effect in EEG. So this difference here. So the incongruent condition, the response goes more negative or just more towards the bottom than this response. So what does this mean? So it turns out this is the moment in which we can actually retrieve the meaning of a word in our mind. So there's a difference here in that the meaning of this word, the congruent word, is said to require less effort to really get to that meaning because it's already been predicted by what came before it. So this N400 effect really speaks to our tendency toward prediction as humans. We have this adaptive response based on learned associations where we can access things more easily based on these kind of sort of predictions or associations that we make. So not as much energy, we could say, is required to retrieve this word, whereas this unexpected ending, we see this more negative response. So again, we can also say this word is easier to process to get to the meaning because of its preceding context than this one. So we see this additional activation needed here. Okay, so while you're thinking about that kind of traditional N400 experimental paradigm and what made those conditions different, I want you to go ahead and picture two different, kind of a different paradigm here where there's two trials. Rather than sentences here, it's going to be something a little different. So let's imagine the participant first saw a picture of a Black man followed by the word violence. And in this case, if we can make the assumption that the word violence is a stereotype of a Black man, let's keep that in mind for this condition. Now let's think about this one, in which a participant would see an image of a Black man followed by the word gentle. So here, if we can assume, again, that gentle is not a stereotype of a Black man, let's recognize that on that basis, this trial could be said to be a congruence on the basis of stereotypes, whereas this one, according to stereotypes, is incongruent. So we still have this kind of setup of congruent versus incongruent between these two trials. So with that in mind, I'd ask you to consider, do you think the word on the left, violence, would be processed more easily than the word gentle on the basis of having just seen a picture of a Black man? If so, then in looking at the EEG response to each of these words, we might see that N400 effect. We might see a more negative deflection of the EEG for the word gentle than the word violent. And if that happens, we could say N400 is a neural marker of implicit stereotyping. It would mean that to some extent, this word had been kind of pre-activated, some sort of association or prediction had been made just on the basis of having seen a picture of a Black man. As it turns out, these kind of stereotyping effects have been shown to exist through N400 in previous studies. So in particular, these studies have looked at N400 on the basis of race, so Black versus white, also on the basis of gender, male versus female. So these kind of studies have looked at stereotyping unidimensionally, so just looking at race or just looking at gender, plus I think perhaps some others. These tend to be the more common ways that stereotyping has been looked at, but again, unidimensionally. Also, it's important to note counselors in training have been shown to have implicit biases, so assessed by reaction time and the kind of designs that I mentioned before. So it seems reasonable to conclude since that group has shown implicit biases, they might also show implicit stereotyping through this kind of study or design. So a great thing about having any kind of measure of biases or stereotyping here is that you can also look at whether there might be any moderators of any kind of stereotyping effects that are found. So in my study, I looked into the possibility of two different moderators based on previous literature. Mindfulness, which is something that's become a pretty popular construct in our fields. So this can be defined as paying attention in a particular way on purpose in the present moment, non-judgmentally. So mindfulness meditation has been associated with less implicit bias. Also a certain aspect of mindfulness, dispositional mindfulness as measured through self-report, has been associated with less implicit bias as well. So another construct, especially relevant when we think about a sample of counselors in training or graduate students in mental health fields more broadly with this topic of stereotyping, is multicultural counseling competence. So this is a way that for a long time in our field, we have thought about training towards more effective work with diverse groups. So this has traditionally been conceived of as awareness, knowledge, and skills. And in particular, the awareness facet of multicultural counseling competence seemed relevant to me. For one reason, this has been previously associated with, it's been negatively associated with racism and discrimination in self-report measures. So with all of that kind of literature in mind, my study in regards to the ERP component in particular, I am focusing on, excuse me, that here in this presentation. You noticed in the manuscript also reaction time was analyzed, but I'm really wanting to highlight the ERP components since I think that might be of most interest. So I hypothesize that graduate students in mental health fields will activate stereotypes as indicated by that larger ERP negativity in the N400 time range for incongruent trials compared to congruent trials, or an N400 effect in this type of experiment. Also hypothesized that mindful observing would moderate stereotype activation such that those high in mindful observing would show less stereotype activation than those low. In the same way, I hypothesized that multicultural awareness would moderate the stereotype activation such that those high in this also would show less stereotype activation than those low. These were the participants. So I did have a focus on counseling students, but to obtain a large enough sample, I extended out into some related helping fields, clinical psych, and social work as well. You can see the participants were mostly female and also mostly white in racial background. So in terms of the methods, participants came into the lab and completed a stereotype experiment, followed by some demographic measures. They then completed the 5-facet mindfulness questionnaire to give that measure of mindful observing, and the multicultural counseling knowledge and awareness scale to give that measure of awareness. So these are some pictures from the lab in which I conducted the study. So this is typically these types of studies, you need a computer on which the participants complete the experiment, and then ideally in a separate room you have a computer on which that data is being recorded, and also another screen that you can actually see the experiment as the participant is seeing it, just to kind of keep track of how things are going. So again, this is a picture from a different experiment, but you can picture the participants seeing the stimuli here that I'll be discussing that they saw and while the EEG data is collected here. And this is just a zoomed out picture of the data collection room. All right, so to give a little more specifics on this stereotype experiment, there were, excuse me, there were 512 trials. As I mentioned before, you do need a lot of trials in an experiment like this to get all those averages. So, we need to see the consistent response within each of these conditions, congruent and incongruent, to really have a valid measure. So across those 512 trials, these consisted of congruent and incongruent trials for 4 different conditions of groups of people: Black men, Black women, white men, and white women. In terms of the words that were seen, these were half positive and half negative in each condition. The words, stereotypes and non-stereotypes that I used, I selected from a validation survey that I conducted with 69 participants to really get the most valid words that most people would consider stereotypes of each of these groups and non-stereotypes. So I picked words from previous stereotype literature, some of it unidimensional on the basis of race, some on gender, and then some of the limited literature that exists on these more intersectional groups, and had the participants select for each word whether the word was, which of these 4 groups the word was most associated with and which it was least associated with, and use that data to create these incongruent and congruent conditions in the experiment. I also asked them whether each word was positive or negative, so I could pick the most positive and most negative words to put into these positive and negative conditions. In the experiment, the participants did have a task. As I mentioned with ERP research, a task is often involved. Sometimes it's more meaningful than others. Sometimes it's just a way to make sure they process what you're wanting them to process. And that was the case here. So participants saw a picture of a person, then one of these words, and their task was to indicate by a button press whether the word they see is positive or negative. So again, this was really just a task to make sure that they read the word. Since the M400 is all about that difference in accessing meaning of the words, just needed to make sure they actually read the word. This is a fuller picture of what a trial looked like. So again, these experiments have to be very precise in terms of milliseconds of when things occur, showing you how long the picture was there, how long the word was there. So that's telling you a little more about that. This is just to give you even more of an idea of what each trial looked like. So it was, they always saw a picture first of one of these 4 groups. They then either saw a word that was a stereotype of the preceding group or a non-stereotype of the preceding group. And that's, again, you can see that's true for each of these 4 conditions of people in the experiment. In terms of my, sorry, I'm adjusting my screen again real quick. Okay, in terms of my analysis of this data, there were, let me tell you about my independent variables. So the independent variables were the prime's race, either Black or white, the prime's gender, either man or woman, and the congruence of the trial. So each one was either congruent or incongruent. There were 2 moderators, mindful observing and multicultural awareness. So I created high and low groups for each of these using a median split. Then the dependent variable, as you might expect, here was an EEG data point. So this was the average EEG amplitude between 250 and 500 milliseconds after target word presentations, that period in which the M400 might exist, where we might see that difference of processing in the EEG. And so this value for each trial, each word, was also averaged across 9 electrode sites. So each of these ERP components that I mentioned, or I showed all the different ones along the waveform or some examples. Each of these tends to show up in certain electrode sites. So an effect is presumably coming from a certain region in the brain that you've isolated, although we don't really have precision about exactly where, since our precision is in this temporal domain, as I mentioned. But still, there are certain electrode sites where an effect tends to be most prominent. And for N400, that's usually in what's called the central parietal region. So I've indicated here, this is the 1020 system of electrode locations, just a standard way that EEG data is collected. This triangle, you can picture this maybe as being someone's nose. So we're getting kind of an overhead view of electrodes. And I've highlighted here the non-electrodes that went into this average that was analyzed in the study. And the analytic approach for this data was repeated measures ANOVA. Okay, so I'm just going to focus on really the more illustrative results here and then talk about everything in terms of the hypotheses. So for Black man trials, there was an average EEG amplitude. There was a difference here. So the amplitude was significantly less for incongruent target words than congruent. So this is an M400 effect indicating stereotypes were activated on the basis of just seeing the picture of a Black man. This is a reflection of the data. So as you can see, I'm describing this as a virtual electrode. This is the average electrode. It's really a kind of an aggregated view of all 9 electrodes that went into this analysis. So it's really a picture of, on average, at each time point, what happened in the data. So it's just a way to kind of visualize the whole grouping of electrodes. So we can think of it in terms of these same ERP images I showed you before. So the thing to note here is, up until this point, I've indicated the M400 time range here. This is what went into the statistics. So this is where we would expect to see a more negative amplitude for incongruent trials than congruent. And as we can see, the red is the average for the incongruent words, so the non-stereotypes, whereas congruent are the stereotypes. So again, this difference here presumably indicates the additional effort that's needed to activate the non-stereotypes versus the stereotypes here. So this tells us that according to M400 kind of logic, that the stereotype content was already active to some extent just by seeing the picture of a Black man. For white woman trials, the same was true. So an M400 effect was found here, stereotypes were activated. So this is the ERP waveform here. So again, we look the M400 time range where we would expect to see a more negative amplitude for incongruent or the non-stereotypes than the congruent or the stereotypes. And again, we see this difference. So this, according to M400, tells us that these stereotype content was already activated by the pictures of white women. These are the table showing the statistics here for paired samples t-tests for each of those groups. So we can see the significant results for Black man and white woman along with the effect sizes. Okay, so in terms of the hypotheses, this tells us hypothesis 1 was partially supported because stereotyping towards Black men and white women was found. Finding something like this does go sort of in line with previous N400 studies that have shown that stereotyping can be indicated through N400, but this study was novel in that I validated these words by combining 2 different groups of people, so race and gender. So in that way, to study this in sort of an intersectional way is novel, so this provides some unique information. And on that note, it kind of provides an interesting perspective in that stereotyping effects were not found towards Black women or white men. So in looking at current literature on this topic, sort of outside of neuroscience literature, since this kind of, it seems that these kind of experiments haven't turned in this sort of intersectional kind of direction, in interpreting this, I find myself looking to the types of scholars that have been considering these topics, stereotyping, discrimination, oppression, intersectionally. So to me, the intersectional invisibility theory is something that, that seems of interest in trying to kind of interpret this sort of novel finding. So this theory really emphasizes potential differences in discrimination and oppression that may be experienced by people with one marginalized identity versus more than one. So, in this case, we can think of on the basis of race and gender, it's the Black men and white women conditions in which one identity is marginalized, whereas Black women would be the condition in which 2 on the basis that I included these in the experiment. And of course, white men, neither, that's the most privileged group. So this theory says that those with more than one marginalized identity may face a more subtle kind of oppression, less overt than those with one marginalized identity. And specifically, this may relate to just a general lack of kind of societal consciousness or really representation that is provided to people with more than one marginalized identity. So within this kind of framework that the neuroscience perspective brings us, this idea of prediction that N400 measures a kind of learned response, combining these different literature areas, it may be that due to that kind of societal kind of lack of representation, that groups with more than one marginalized identity, in this case, Black women, these stereotype content may not exist. And that kind of, in a way, is maybe an unfortunate reflection of a lack of representation that's provided, since this can be thought of as sort of a learned response, according to N400. In sort of a similar way, that kind of thinking might help us better understand why white men would not be a group in this experiment towards whom stereotyping is shown, just due to the degree of privilege that white men usually have or do have, and really perhaps on that basis, the wide variety of different representations of white men that may exist that may not contribute to sort of a learned response or kind of hardened sort of expectation of how white men might be. In terms of the hypotheses, so neither of these related to mindful observing or multicultural awareness were found to be true. So in terms of mindful observing, it may be that mindfulness related effects on implicit bias that I mentioned do exist, may relate to emotion processing, since bias is itself an affective construct. So it's considering our tendency to feel a sense of positivity toward a certain group or feel negativity towards another. Whereas in this study, as I mentioned, stereotypes can be positive and negative. And in each condition, I included both positive and negative words. So I was really kind of controlling for the valence of the stereotypes. So that might explain why mindfulness didn't relate here. In terms of multicultural awareness, not moderating this, this may illustrate differences between explicit and implicit measurement, which I was discussing earlier. So it may be that in a sort of explicit measurement situation, like how we measure multicultural competence through self-report, that these kind of controlled processes may be obscuring sort of automatic response that may be there. And that was exactly what was measured through implicit stereotyping here. So this may illustrate really a utility for us of being able to measure these constructs implicitly, or automatic constructs. It may also be that the type of bias that was assessed by the multicultural counseling knowledge and awareness scale may be a little different than the type of specific stereotyping tendencies that were found in the experiment, specifically towards Black men and white women. Okay, so some implications of the study. Since automatic stereotyping was indicated, I mean, it should obviously make us think about the fact that if this does exist, it could influence the relational type of work that we do in counseling. We need to therefore increase reflection on the possible influences of this type of automatic sort of processing in our work with clients, both our sort of work in the moment, as well as even our conceptualization of clients, thinking about ways in which these sort of associations may influence our thinking and acting towards clients. We should also think about how neuroscience methods and the kind of conceptual frameworks that inform them that I've been talking about here with N400, you know, this idea of kind of prediction, how might this more broadly help us promote a social justice orientation? So does this better help us appreciate the potential influences of these kind of human tendencies in our work with clients as well as in the real world? So, you know, I think it can be meaningful to ask students to consider if these kind of tendencies exist, even in people who are being actively trained in mental health fields to work with diverse groups. What sorts of effects should that then lead us to consider may be happening in the real world among people who may have no impetus to consider the sorts of biases and stereotyping tendencies? Instead, they may, you know, just kind of function and see the world through those. So I think that can help us really promote more of a social justice orientation in our field, which is certainly something we're interested in. Also, the study, I think, indicates implicit stereotyping seems to show unique patterns when we kind of combine categories, which hasn't really been done, it seems, in the past, so that can be important in future research. Okay, so in terms of future research, I think that we do need to examine whether you know, these types of methods and discussions of them can promote greater reflection on stereotyping and biases in our field and in our students. And again, to consider whether what other kinds of ways that neuroscience methods and frameworks can improve counselor training and counseling practice, and to design more studies that do look at stereotyping and biases intersectionally. Also, to really expand this out into other groups who have not really received treatment in the research literature. So, one reason I thought it was helpful to kind of move in a direction of combining categories is because they have been treated unidimensionally, but by the same token, there are many groups that we haven't really explored stereotyping or perhaps biases towards. So we need to continue expanding that literature. And I think it can also be helpful to, even though effects were not found in terms of moderators here, to continue exploring that in terms of whether these kind of tendencies can be moderated by anything in counselors. Also, to step back even further, I certainly have an interest in this kind of research, and the study that I've presented to you have increased my interest in other uses of these types of experiments. So, through using an ERP method, for example, that has such a rich history and literature of being able to isolate certain neural markers and certain experiments, I think this could give us a lot of power to really kind of bring the brain and this kind of perspective into the equation in terms of assessing client symptoms and even some symptom reduction through counseling interventions, and could really give us a lot more knowledge in that regard. Okay, so this kind of study, in my experience, and also my experience of working with others who've done this kind of work, it's collaborative. There's typically a lot of people to thank, and I certainly have people I want to acknowledge here. Some mentors at Alabama, both in neuroscience and counseling, and also the lab members where I collected my data. Day to day, we would help each other with all sorts of things that would come up in terms of getting our projects and all off the ground. And also want to thank the Alabama Counseling Association for a research grant that supported this project. So with that, I'm really happy to take any kind of questions or comments that y'all have. Thank you so much for letting me present to you today.
Thank you so much, Dr. Weber, for such a rich and fantastic presentation. You can see in the comments, people also express the same sentiment. That was just so, I would just have to ask you, after doing this research, in what ways has it impacted you personally as a professional, and how have you changed after doing all this?
Wow, great question. I certainly think it's increased my curiosity. I guess I see such a level of depth in this, using these kinds of methods, and it's only increased my interest in kind of expanding the methodologies that we have access to as counselors. I guess coming from that perspective, I mentioned early in the presentation of where can our counseling values and interests take us in terms of methodologies. Using these kind of things, these kind of methods can feel maybe foreign to us, but I've really taken an interest in kind of making sure that the things we do are rooted in our own professional values, but kind of seeing where that can take us. And that's a way that I really framed kind of my journey into neuroscience that I described early in the presentation.
Thank you. That's great.
Yeah, I'm seeing there were a lot of chat items. I hope those weren't. I didn't have access to them when I was presenting. I'm happy to try to get through them if you'd like.
Yeah, let me see if mostly just people sharing how important what your entire presentation is, and just expressing gratitude.
Oh wow.
I'm trying to see if there's specific.
Oh, I had one I could pipe in the meantime. So given the EEG wearables are becoming so much more commonplace, there's so many more of them. Are there any that you like in particular that you would think counselors, we could use them in session today?
You're exactly right that I've noticed myself also. There's a trend towards making things, I guess, more practical. For example, like EEG that doesn't even require gel. I know that can be a real kind of impractical sort of thing, but yeah, I feel like my training was so rooted in traditional types of stuff that I probably wasn't able to pay as much attention to that sort of thing as I could. But I know there's definitely a lot out there, and I understand definitely the rationale and like moving towards things that that are more practical. For example, I didn't mention in my presentation that this type of research in our lab, a lot of types of studies that you do, you would need like an hour, no more than an hour with people. In our case, we reserved an hour just for setup in terms of getting the EEG ready and that kind of thing. So, you know, that there can be this distinction between the lab and the real world. And I do think there are people who are trying to bridge that, which is what your comment reminds me of. And I certainly have interest in that. But I guess to this point, I was in such a kind of lab-based mindset that I wasn't able to look into that. But absolutely, it's an interest of mine.
And Dr. Weber, I think you did a wonderful job expressing the need for rigor and validity in any variable that we measure, whether it's a self-report variable that comes from a Beck depression inventory or a self-report variable that comes from the scalp, you know, and just being able to really assess the quality of any variable that we have. And I think when we look from like consumer grade materials, you're going to lose some of the validity in the measurement. There's going to be a bigger time delay. So when you look, one of the benefits of ERP, as I have learned even more now through Dr. Weber, is the precision and the timing, you know, and you won't get that from a consumer grade wearable device, at least not right now. As maybe there's other ones that are out there that I'm unaware of, but it's all about the number of samples that you take, the integrity of the reading. And what makes it hard to do EEG in counseling is the interference that Dr. Weber talked about of, you know, when I move my jaw, it affects the reading. When I talk, it affects the reading. So then we look at other things that are less, but I don't know, I'm rambling. Dr. Weber, what would you say to some of those, I guess, threats to validity that would exist in lab research versus direct clinical application?
I think it's exactly what you just highlighted. I think the real challenge is it's kind of what I was illustrating and going from my interest to begin with, which has a very real world sort of feel as I guess it seems that currently our way of working out into the real world is to interpret these kind of studies and like, you know, have great theorists who can become knowledgeable of all these lab-based studies and kind of put together hypotheses about the big picture. And then we sort of picture that out in the real world. But to your point, it's hard for validity reasons to really validly get out into the real world and kind of see these things in action for all of these, you know, sort of confounding types of reasons. You know, it doesn't mean that it doesn't happen. You know, part of what I've seen in trying to, you know, design my own studies and try to establish a certain thread among different, even lab-based studies, is these methods are still evolving even in the lab. So, you know, the kind of the idea of putting a label onto something that you saw in a study really needs to be replicated over and over to really see that what you are kind of assuming is true, because just because, you know, it requires such precision that I think over time we're getting even better at, but you know, it's still such a new kind of endeavor, I guess. And that's really what led me to appreciating the kind of validity you just described, is kind of knowing that what you're saying has a foundation in previous studies, and ERP to me has really done that in a way that, even though it may seem off-putting at first to kind of see the way it's set up, when you really want to make sure you have a good sort of lit review and understanding of these effects, it lends itself to that.
Yeah, if I could jump in there just for a second. Wes, you know, I always have high expectations of you, and you just exceeded them again. I'm sitting there in awe, like, wow, this is really complex stuff, and you really explain it really clearly. I have to walk back through my mind going, independent variable, dependent, and you've got this down. It's so smooth. I was going to say, just jump in there with Carmen's question, which I think is a really good question. There does seem to be this as the precision goes down, the claims can go up in consumable or consumer wearables and in other devices, because there seems to be more investment in marketing than it does validity. And that's, I think what you presented today is just something that we can keep in our pockets when our clients are coming to us and saying, hey, I got this new thing that's going to help my depression. The analog I use or would use is that, does anybody have one of those massage guns that's all the rage now? I jumped on the bandwagon, but like suddenly now all the things that we knew about stretching and muscle maintenance is out the window. You just need this vibrating gun to pound away the problems. And I think that's just sort of a parallel here that you're bringing us back to. Therapy is messy, and so is EEG and ERPs, all of that. So I appreciate you bringing it back to us, and Carmen, I appreciate the question.
Yeah, Chad, those comments really resonate with me. It's that thought probably is why I chose to express, like, I expect my starting point that I described of like theoretical work, conceptual work is probably a lot of our starting point, because that's the stuff that literally is kind of giving us, it's putting the brain in the world that we already exist in clinical work, real interactions and stuff. And, why wouldn't we start there? That's where we live. But then I guess, I have developed this appreciation through learning about these methods for, why do things happen in the lab, because at this point that tends to be the best way to really establish validity, and kind of when it's your job to create a study that you need to justify and that you want to make a hypothesis that might actually turn out to be true, it becomes important that you have those studies to really make you confident. And I do think there are methods being used right now that I found wouldn't give me that confidence, and I do think a lot of times they do tend to be something that's a little less lab based.
I told you I'd nerd out, and so you all stop if I ask too many questions today. But this may be my naivety, but also I think it's a validity question too. With some of the implicit bias research, is there a way to tease apart what somebody might, like, I cognitively know that there are stereotypes attached versus I feel and believe in the stereotype attached? Can you tease that apart with ERP research, or would the response look the same whether I have a cognitive awareness that this stereotype's there versus like, I believe that's true. I believe Black men are violent versus I know some people believe Black men are inherently violent.
To me, when I hear that, it's like I don't have an immediate response, but I immediately start thinking like, that's where you tweak an experimental design, or you add a measure, or that kind of thing, because that's exactly how this kind of literature kind of evolves. If you can use what's existing, whether it's either in the behavioral self-report domain, or if you find a way to maybe add a condition or something within an experiment, that is how you can kind of add on things. But to me, it's a feeling of creativity. Maybe back to, I think, Emily's question of how I've kind of changed. It's like this kind of stuff becomes a sort of a mixture of validity and tradition, but also creativity in terms of, you develop new questions and you want to be able to pull from existing literature to kind of look into that, but in a new way. So I can't say that I know of a way to do that, but it's fascinating. I'd love to keep thinking about it, and probably will. So thank you.
So this study itself distinguish between those two. It could be that the respondents knew of those stereotypes, or it could be that they felt those stereotypes. Is that accurate?
Yeah. So let me kind of think about, again, in these terms, in 400, what this sort of indicates. And there are different debates you find even about a certain component as to like what's the best words to put onto it. But kind of minimally, this kind of design would say that just on the basis of seeing a picture, you've activated certain associations, including the content of the word that's about to be presented, and then you verify that through this N400 effect. So some of the more, I guess, trying to bring this into the real world, which again is all of our interests, the more kind of maybe provocative kind of claim would be, does this happen in our real life? Do we see someone and we activate this sort of expectation that may or may not come to inform future thoughts or whatever, but it happens in some way that this experiment sort of shows. So yeah, your questions about maybe exactly kind of more nuances of what exactly does that mean may not be completely clear, but maybe could become more clear in time.
Yeah, I love it. And that's the joy of being curious and asking more questions and adding new conditions. So that's where my mind was going. I was like, how would I respond? Because I know these things I know these stereotypes exist. Do I know that I believe them? And maybe it doesn't matter. Like maybe knowing that they exist influences us at a deeper level, and ERP can show that, just simple awareness of stereotypes sometimes can influence bias responses, whether you believe them or not, or whether you consciously say that you believe them or not,
Right.
Which is probably why early multicultural counseling education courses that taught about stereotypes may have not been the most helpful back in the day. And now we're moving away from stereotype education to something deeper.
Mm-hmm.
I appreciate everything you brought today. Thank you.
Thank you so much for the questions, Eric.
I have
Anyone else? Yes, please.
maybe kind of a crazy question, but if we have a client who wants to work on their own implicit bias, they're either aware of it, as Dr. Beeson said, in a general way, or they may even suspect or be aware that it's there. Other than the mindfulness, do you think that cognitive therapy or something like that might actually make a difference for them?
Yeah, and I certainly want to open this up to others. I think this is an area others could probably contribute as much or more than me on that question. But yeah, to me, I'm thinking about that sort of distinction between kind of bottom up and top down sorts of change, which to me became my interest in mindfulness, is more of this maybe bottom up way of changing. Perhaps even my thought here was that maybe people who are more mindful don't develop the kind of learned responses that were shown in the experiment, just ways that they are present in the world or that kind of thing. But yeah, when you mentioned cognitive therapy, I don't know exactly what you mean in terms of what that would look like, but I guess it's more of deliberately kind of thinking about your biases and trying to maybe refute them. Is that what you're thinking?
Well, a couple of things. I really agree with you about the mindfulness. I also believe maybe it's more true with people who are older, but the messages that we've gotten since we were born, through every media and just it's overwhelming what contributes to these biases. And I think I read about a study about how certain counseling students who were tested for implicit